Hi guys, I'm Rachit from BSMS Physics, and my project today is WBC segmentation and classification. So, project overview. Our problem statement is that the manual examination of blood samples under a microscope is time consuming and prone to human error. Also, specialists aren't available 24/7 throughout the country. The solution to this is to develop an AI based system that can automatically segment and classify white blood cells from microscopic images. Why does it matter? This project enables faster and more reliable disease diagnosis by automating the process of white blood cell identification and classification. It can help us detect diseases like leukemia and infections. Project objective: to develop a deep learning model that can accurately segment and classify white blood cells into their respective types. Uh, these types being eosinophil, lymphocyte, monocyte, and neutrophil from microscopic blood images. This classification aims to assist in automated blood analysis and disease diagnosis by recognizing the distinct morphological features of each cell type. So, what are what are the processes involved in this? First, we gather white blood cell images from Kaggle and then create segmentation masks to highlight the regions of individual WBCs. Second, we train classifier, which is we basically develop a CNN based classifier to categorize these segmented WBCs into four classes. Uh, like the one mentioned above, eosinophil, lymphocyte, monocyte, and neutrophil. Uh, the second last step is to build a segmentation model. We'll design and train a unit-based architecture which will accurately segment WBCs from background in microscopic images. Lastly, we'll combine the unit segmentation model with CNN classifier to create a system capable of detecting, segmenting, and classifying WBCs. So we should understand what segmentation and classification mean. In my project, I begin with classification using a CNN to identify different types of white blood cells. Since there are multiple categories, this is a multi-class classification problem. The CNN learns unique visual features from the image of the cells, like shape, texture, and color, and then classifies each of the image into one of these categories. So, um, segmentation in machine learning is the process of dividing an image into meaningful regions, so that each part corresponds to a specific object or area. It helps the model understand where the objects are located within an image and which pixel belong to each object. So here is an example of instant segmentation. So there are two people walking, and these are cars. So the background has been completely separated from the main objects here. Um, some more about segmentation. In my project, I've used instance segmentation, which not only identifies the object class like white blood cell, but also separates each individual cell as a unique instance. Even if multiple cells belong to the same category, this will give a more detailed and accurate understanding of the image. Compared to other types of semantic segmentation labels, all pixels of the same class together without distinguishing between individual objects. For example, all WBCs would be marked as one single region. Uh, panop- uh, so, one of the types of segmentation is panoptic segmentation, which combines both semantic and instant segmentation, providing both class of each pixel and individual object boundaries. In contrast, instant segmentation focus specifically on detecting and outlining each separate object's instance, which is ideal for medical images where individual cell boundaries are important. For example, here we have a lymphocyte, and this is the segmented image which has been outputted by the mask to PNG file. So yeah, so this is so we must understand about CNNs. A CNN is a special type of neural network that is really good at understanding images. It can look at a picture and figure out what's inside. It works by automatically learning features or patterns from shapes, such as edges, corners, textures, without needing manual feature extraction. The core idea behind CNNs is the convolutional operation where small filters slide over the image to detect local patterns. The core idea behind CNNs is the convolution operation, where the small filters, also called kernels, slide over each image to detect local patterns. Each filter learns a specific type of feature, like we discussed before. After convolution, the ReLU activation function is applied to introduce nonlinearity, helping the model understand more complex patterns. So what is ReLU, right? So the function will give some output based on some weights and some parameters, right? So the what ReLU function does is when the output is below zero, it will give an output of zero, right? But if the output is greater than zero, it will give the normal value of the output, right? So basically all values below zero turn to zero. That is basically what ReLU activation function does. Next comes the pooling layer, which reduces the image size by summarizing the most important information, making the model faster and more efficient while keeping the essential features. So here we can see this is the kernel, which will extract certain information of a certain part of the image, right? We'll proceed with the convolution layers and ReLU activation function. After this, we'll proceed with pooling, different pooling layers, right? What pooling does is, for example, in this case, we have used max pooling. So let's say there is a 3 by 3 kernel. It will take the maximum value out of that 3 by 3 grid and associate the value of that particular pixel in the next convolution with the maximum value found in the kernel. So after pooling, it will flatten like the outputs, the different layers, and then the neural network will help us identify what category does that image belong to. So it will be a probabilistic distribution, and whichever category has the highest probability will be the one we'll be outputting. So onto the main architecture behind my project, which is UNet. UNet is a deep learning architecture mainly used for image segmentation, es- especially in medical image analysis. Its goal is to precisely identify and outline objects within an image, such as cells, tissues, or organs. The model gets its name from the U-shaped structure, which consists of two main parts, an encoder and a decoder. So this part is the encoder and this part is the decoder. Also, there are skip connections between the encoder and decoder so that when the decoder is reconstructing the image and the final mask, it is able to preserve some of the data which was lost during this process, right? So what is the working of UNit exactly? The encoder, which is the left side of the U, works like a feature extractor. It uses convolution and pooling layers to gradually reduce the image size while learning important features and patterns. The decoder, which is right side of the U, does opposite. It unsamples the compressed information back into the original image size to reconstruct the segmented output. A key part unit of UNit is the skip connections, as I said, that link encoder to its corresponding decoder layer. These connections help uh, model retain fine spatial details that might be lost during downsampling, resulting in more accurate segmentation boundaries. In simple terms, UNit first understands the image in depth, which is encoding, and rebuilds into the detailed segmentation map. So I'll be showing you what my code looks like. So uh, first we have started the environment and we have given a random seed, right? We have given it a path which we have allotted the path to our data set, namely our segmentation data set, which are our masks and images. So first we have the convolution block, which will be used again and again and has been defined as a function. So we have a 2D convolution. We then apply batch normalization and ReLU activation to it. So lastly, we'll apply a dropout so that the model is able to generalize better. We'll drop out some of the features so the model can generalize better. Yeah. So we have our encoder block, decoder block, and we put four encoder blocks, then a convolutional block, and four decoder blocks, which helps us map that um, unit architecture. So we have a training and validation data set. Um, this uh, here I have made use of uh, in data set augmentation because my data set sample size is a bit small, right? Hardly 100 to 100 images. So I have made use of image augmentation and I have flipped the images right, left, up, and down, right? And same I have done with the corresponding mass. So I have trained that model, unit model, and you can see that at the end it will give you like an accuracy of 98 something percent. Um, the other thing is this is my final code. So I have made a classifier and I have given assigned the weights and the biases to a specific file, namely classifier.joblib, right? And I load that model here. So um, this is like one of the results. So this was the image sent from monocyte like data set, and then it was able to correctly classify it as monocyte and give the mask, which is like these, right? So it was able to properly segment. Um, so here are the metrics. The unit architecture gives you an accuracy of 99.14%, a precision of 99.2%, and a recall of 99.1%. The classifier, however, is a little less accurate. It gives you an accuracy of 96.88%, a precision of 96.2%, and a recall of 97.5%. So, yeah, what did I learn from doing this project? First of all, this project gave me a hands-on experience in Python libraries like NumPy, TensorFlow, CV2, Matplotlib, and I also learned how to handle images in Python. Second, there were some problems I was facing while creating datasets for image segmentation, right? So I could not find suitable cell masks. So I manually created the segmentation mask using Make Sense AI, and I did it for like 200, 300 cells, right? And with help of image augmentation, like I described before, I was able to expand that data set into like three or four times. So this was one technique that I personally utilized, and it allowed me to generate accurate black and white mask data for training. I used image augmentation techniques to expand the dataset and introduce greater variety. Finally, I would like to talk about the unit architecture. Right? I was able to understand and implement the unit architecture from the unit research paper. Thank you.